Hello, today I will talk about the models of the cell membrane. How first, Davison and Danieli proposed a model in 1935 and how it was later proven wrong, well, kind of, by Singer and Nicholson in 1972. So let's start. Look at this. Looks like an ice cream sandwich, doesn't it? Well, that's because it is. I'm kidding. This is the Davison and Danieli model, which proposes a phospholipid bilayer surrounded by two protein layers. No proteins in between. However, this model is off. I know. But who could have blamed Davison and Danieli? And if we take a close look, it is not that different from the model that we use today. But what proof did Davison and Danieli have to suggest this as the cell membrane model? Number one, in an electron micrograph, they saw two dark layers and one light layer in between. I'm gonna insert a picture right here, which might have explained that proteins and phospholipids were placed that way. Two, the amount of phospholipids in a red blood cell was twice the amount of a regular cell membrane, which explains the phospholipid bilayer. 3. Despite being very thin, the cell membrane is very effective blocking substances, which is why they thought that it had proteins. They were a little off as to where those proteins were, but that's okay. We'll unwrap that later. Now, how was this model proven to be wrong? We'll see. 1. Singer and Nicholson found that proteins are different in size and in shape, unlike here. 2. Proteins have a hydrophobic region? What? All of them can't be attached to the surface. You know who doesn't have a hydrophobic region though? The heads of the phospholipids. 3. They used a technique called freeze fracturing to crack open the cell membrane. And what did it have inside? Bumps. Bumps? What? What were those bumps? Protein bumps, which means the proteins were not completely in the surface, but they had to be stuck in there somewhere. 4. They used another technique called fluorescent body tagging to tag these proteins, and they realized that they moved laterally through the membrane, all over the place. So what did this mean? It meant that it was contrary to the Davison and Danieli model. So they had to do another model which accurately showed where the proteins were placed in the cell membrane. And thus was born in 1972 the Singer-Nicholson model. Surprise! And we will build it together, shall we? Okay, so of course you need your phospholipid bilayer, which was proven to exist in a cell membrane. But what's gonna be different here is the proteins, which will not be up here, but in here with the phospholipid bilayer. Pretty cool, huh? This is an integral protein, but we also need other proteins such as channel proteins, so we can let some substances through. Here's the little hole. We also have peripheral proteins, which are not completely in the membrane, if you know what I mean. And let's add a glycoprotein there. Why not? Here is an accurate representation of the Singer-Nicholson model. We did it! I hope now you understand why we use the Singer-Nicholson model instead of the Davison and Danieli sandwich model. Guess it was just too cheesy. I'm sorry.